All right, here we go. This is part one of my new chapter by chapter walkthrough through the Endogenous Retrovirus Handbook. Now, for anybody unfamiliar with which book that is, exactly right here, the Endogenous Retrovirus Handbook, dismantling the best evidence for common descent or common ancestry. So I've written an extensive book on this topic. A big reason as to why I've chosen to spend so much time and put so much energy into the topic of endogenous retroviruses is because the evolutionary community looked to shared ERV sequences between various biological organisms, specifically humans and chimpanzees and your other primates, as the single best line of evidence for evolution. And so if they believe ERVs are the best line of evidence for why they believe humans and whales and strawberries are related through common ancestry, then I want to make it my mission to dismantle that argument. And so in 2023, life is busy. People are go, go, go. And so I understand people, including myself, we love podcasts, we love videos, we love audios, audiobooks, for example. And so I thought, you know what? I am going to do a new series going over every single chapter. And so it's going to be broken up into parts. Some chapters are very long and extensive, so I'll probably have to break those up into several parts. For example, the final chapter, I'll probably break that up into a few parts, especially because some of the chapters are very technical. And rather than doing a very strict audiobook, and so I did an audiobook about a couple of years ago on the Independent Origins Handbook. And so that was very strict. Matt and I edited it. We made sure there was no mistakes. It flowed nicely, no commentary, just straight up audiobook. And it did well. It is available for free to the public. You can find it on the YouTube channel itself. You can also find it on the website under resources. You can watch, or I should say, listen to that entire audiobook. Again, that's the Independent Origins Handbook. For this one, rather than being a strict audiobook, it's going to be more casual. And so it is going to be a casual reading of the Endogenous Retrovirus Handbook, and I will be providing a lot of commentary. I will be providing a lot of feedback. I will be adding in details and expanding on what I am reading. Most of the commentary will be at the end of each reading session. So, for example, this episode is specifically going to be going over the preface, basically, okay, or the preface of the Endogenous Retrovirus Handbook. And so after each reading, I will go over I'll go over some commentary, add some details. And so I want to share screen real quick. I won't be doing this long of an intro before every episode. I figure since this is episode one, I'll give everybody a little bit of details. Now, one thing to point out is this is going to be Patreon specific. And so for those that are patrons, those that are financial backers or partners with the Standing For Truth Ministry, they are going to get first access to each part, to each episode of this series. Now, for as little as $1 a month, you can become a Standing For Truth patron. We are simply looking for more Standing For Truth Ministries partners in order that as we move into 2024, we have more support for the ministry. And with more support for the ministry, we can get more guests, we can get more big debates, we can get out our books a lot faster, our videos. Also, it helps us spend more time and be more diligent in our active research projects. We'd also like to, in 2024, do some in-person conferences, some in-person panel discussions where we can get together. Of course, that is all costly. And so if it is on your heart to support the ministry, again, this is one way to do so. You can become a Standing For Truth partner simply by donating or providing a gift through our website, through our PayPal specifically, and also just by becoming a patron. Now, for this series specifically, to anybody who is a patron, to anyone who is a financial backer, please do reach out to me, and I will send you a free PDF of the Endogenous Retrovirus Handbook, and that way you'll be able to follow along in this series as I go through every single chapter, word by word, sentence by sentence, paragraph by paragraph, and we're going to leave no stone unturned. This is always also going to help me as I work towards expanding the Endogenous Retrovirus Handbook, as there are critiques that have been put out, and I am thankful to the critics of my current work on ERVs, because their critiques are a gift to the separate ancestry model and creation science, because the criticisms, the challenges, the objections only help to strengthen my specific model on explaining the existence of endogenous retroviruses. And so I am planning on expanding the book greatly as I work towards addressing criticisms and challenges, and also expanding my model. I'm about model building, but I'm also about refuting the best so-called lines of evidence for evolution. And so in refuting evidence for evolution, we also model build. We show why the separate ancestry model, or young earth creation in general, can explain the data, in this case, endogenous retroviruses, much better than the common descent model, much better than the evolutionary model. Now, I believe this series is important, and I want as many people as possible, especially those that are serious about the creation versus evolution debate. I really want to encourage you in becoming a Standing for Truth partner or patron to gain access to this series, because we host a lot of debates. We engage with a lot of critics, including the most militant critics online and in the world, really. And they advance endogenous retroviruses as the best line of evidence for evolution. As a matter of fact, they will oftentimes point out that the existence of shared endogenous retroviral sequences in the biological world is irrefutable or undeniable evidence for evolution and against special creation. And so I want my brothers and sisters, I want the body of Christ to be equipped. I want them to be equipped in how to defend separate ancestry or biblical creation, and how to just decimate, to absolutely demolish endogenous retroviruses, and also the other so-called best lines of evidence for evolution, like nested hierarchies, junk DNA, pseudogenes, chromosome 2 fusion, and so on. And so I do believe that walkthroughs of our books like this are important. After I'm done with the ERV handbook, I will be doing a chapter by chapter walkthrough through my special creation book, which just touches on a ton of topics. That book is huge. That book is over 330 pages. And so that'll be quite the project. Okay, so real quick before I get into the first chapter, the preface again, I just want to show everybody where they can pick up a copy of the book. Of course, if you are a patron, reach out to me. I will send you a free PDF of the Endogenous Retrovirus Handbook in order that you can join me in following along as I do the walkthrough. So right on the website, Endogenous Retrovirus Handbook, you click the link. We got a black and white edition, full color edition. Currently, the black and white edition is actually the limited edition, the orange. And people love this edition. It sold a ton for a while. We were number one bestseller, number one newly released bestseller on Amazon. And partly due to the limited edition. People love it. I love it. And so it seems like it's sticking around for a while. But you can find that by taking the link to the Amazon page. You can also find our other books, of course, the End Times Theology book that I've written, and then also Special Creation. And so you can see here on the website the debates that I've did on endogenous retroviruses. So what I like to do when I write a book, and especially release a book, is engage in, several formal debates on it. I want to show the world that I can put out a book that took a lot of work, energy, required a lot of research. Studying these topics is basically a full-time job. And so your support helps people like myself study these technical topics full-time and then convey my conclusions and my research to you guys as we demolish evolution and defend the truth of biblical creation. And so I love engaging in formal debates. And I have, as a matter of fact, engaged in formal debates with critics on every one of my books. Luca Medugno, he's a chemistry teacher, very knowledgeable. He's a chemistry teacher in Italy. He got my Endogenous Retrovirus Handbook probably over a year ago. And he read through it, thoroughly studied it, put together what he believed were criticisms, challenges, objections, so on and so forth. And then we engaged in a formal debate. And I think that went incredibly well. So I do recommend people check that out. I've also done plenty of lectures and given plenty of presentations on endogenous retroviruses. So if you do click the video section, go down to endogenous retroviruses, and in the endogenous retroviruses section, you'll find a list of the lectures I've given associated with the endogenous retrovirus topic. So all things ERVs, put it that way. And there's a series here that you can check out, including some debates. So with all of those details and that introduction out of the way, we are going to get into part one of, again, a chapter by chapter walkthrough, specifically this walkthrough focusing on the Endogenous Retrovirus Handbook. And so let's get right into it, everybody. Table of contents. Briefly, I'll go over basically the structure of the book, what we have in terms of chapters, topics. And so we start off with the preface, okay? And then we go into the introduction. So essentially, I just talk about what to expect in the book, briefly go over what endogenous retroviruses are, why I find this topic important, why I find engaging the critics on this topic in detail is important. Then I get into the important details. That is chapter one, basically going into what are ERVs, what about ERVs the evolutionist believes makes for good evidence for common descent. Again, the structure of ERVs, how they're built, essentially with LTRs, long terminal repeats on either end, ENV, GAG, and pole components, or genes within the middle of the ERV. We've also got what's called solo LTRs all throughout the genome that you'll see as we work through this book, and I also provide commentary, are not necessarily the ancient remnants of past retroviral infection, nor are they degenerated endogenous retroviruses. They may simply represent functional stretches of DNA. Then I get into chapter 2, design diversity. This is an important chapter that talks about the biblical model, but specifically the design diversity hypothesis and how endogenous retroviruses and other classes of retrotransposons apply to this model and starting point, that God would have designed or created the original kinds with internal diversity. So rather than being homogeneous genome-wide, created organisms would be heterozygous. This goes back to basic genetics, where alleles, capital A, lowercase a, capital B, lowercase b. Okay, if you're capital A, lowercase a, you're heterozygous, and there's DNA diversity there. If you're capital A, you're homozygous. There's no DNA diversity there. So if you apply this logic or this starting point to millions of positions within the genome, where you've got capital A, lowercase a, capital B, lowercase b, you have almost unlimited potential for genetic variation. Every generation, DNA variants recombine to form new chromosomal combinations. And You've also got front-loaded in organisms many DNA elements, such as endogenous retroviruses, allele sequences, so-called pseudogenes, which we now understand are necessary to sustain healthy life processes in the cell. And so all of these DNA elements apply to the design diversity because they are various classes of DNA, functional DNA elements that were front-loaded in the beginning, that are there for adaptation, they're there for change, they're there to benefit creatures, and even for natural selection to act upon. Because natural selection is a fine-tuning mechanism that keeps a species as strong as it can be. And that has nothing to do with evolution, doesn't add anything, but it can help in terms of organisms adapting to various environments. And as we know, adaptation is not large-scale evolution. And so that's an important chapter that goes into the, or at least the creationist model that I hold to, the starting point and how all that works, including predictions based on that starting point. Then we get into the non-discriminatory evidence. That's chapter 3. And by non-discriminatory evidence, we mean homology, right? Shared structures in the biological world. Also nested hierarchical patterns and so-called transitional forms. Those lines of evidence that are agnostic to the overall debate. What I mean by agnostic is if I were to say to an evolutionist, creation's true because the sky is blue or the earth is round. Well, the evolutionist would say, what are you talking about? We can also explain why the sky is blue and the earth is round. It's agnostic to the debate. Many different models can explain why the sky is blue. Okay. One example, homology, which I touch on in this chapter. I'm just going over the details of each chapter briefly. Homology. The creationists would explain homologous patterns as being the result of common design. Human engineers build in homologous patterns. And so why shouldn't God? Evolutionists would explain homologous patterns, again, shared structures in the biological world, as being the result of common descent, that these structures were inherited from past common ancestors. So we need to look to discriminatory lines of evidence because there is a lot of overlap in the creation versus evolution or origins debate. And so we need to focus on those lines of evidence that can discriminate between the models, that can help differentiate between the models. And one discriminatory line of evidence that I put forth in this book has to do with DNA function. And that's where we get into chapter 4, which is titled Created Units of DNA Function. Essentially everything about the first 3 chapters, including the introduction and the preface, sets up what is contained within a very detailed chapter, chapter 4. That's where we really demolish endogenous retroviruses by utilizing the conventional literature. There are dozens of papers in this book. And so if you have the physical copy or the PDF, you will see all of the sources when I cite them, and you'll see that the book is packed with sources. And then we get into a conclusion, common descent overturned, and then, as usual, an about the author section and a references section. And so with that, we're going to get right into the first part of this book, the preface. Okay. By Donny Badinsky. Why did I write this book? I wrote this book because as a biblical creationist, I find it incredibly important and enjoyable to deliver the most powerful and well-researched responses possible on the topic of creation and evolution. These responses are to the best lines of evidence proponents of universal common ancestry can offer. I have examined the question of ancestry, universal versus separate, intensely. In addition to dismantling the arguments for common descent, it is my objective and my responsibility to offer a superior model. Since the truth is so important, it is also my purpose to communicate this valuable information to as many people as possible. and to convey it in a way that is comprehensible. I want people to feel the same joy that I felt when I came to the knowledge of the truth. Deep time evolution is not the truth. The truth is far greater than common descent. And it turns out that the scientific evidence is extraordinarily consistent with the Bible's account of origins. I have thoroughly examined the evidence, and it does not support evolution. This book provides the reader with the highest quality arguments and data available. The reader can be delighted to know that I wrote this book with the goal of leaving no stone unturned. The arguments presented are supported by the scientific literature. The question of ancestry has been answered, and the answer has massive implications concerning our origins. I have had the privilege of hosting and moderating a myriad of debates, 250 plus, as of February 2023. As a matter of fact, now, and this is my commentary, we as a ministry have hosted, as of now, August 2023, roughly 330 debates on all sorts of topics: soteriology, eschatology, which is end times theology, soteriology being the doctrine of salvation, creation, evolution, debates on atheism, right? Atheism and naturalism, debates on dispensationalism, all kinds of debates. And so do check the channel and the website for those debates. Okay, continuing on. I have also had the honor of interviewing some of the most prominent and well-informed biblical creationists in the world today. I am grateful to have hosted so many interviews, lectures, and conferences connected to the origins debate. The many scientists and apologists I have met in doing full-time Christian ministry and apologetics have done a phenomenal job defending and upholding the truth of God's word. I have also defended the truth of biblical creation in numerous debates and discussions. As a matter of fact, I have had more than 95 live debates as of February 2023. I will add, again, this is commentary, as of August 2023, I have had my 100th formal debate. And so since my 100th formal debate, I've also engaged in podcast debates, endless open mic debates, endless impromptu debates. But formal debates, I've now had my 100th formal debate. That was a few months ago. And so rather than 95, I am up to 100. I have been confronted with many of the best arguments the skeptics have to offer. In my years engaging with the critics of biblical creation, it has become clear that the existence of endogenous retroviral sequences found across the biological world is regarded as the best evidence for evolution. I have personally engaged the most militant of evolutionists on many occasions regarding this topic. And people need to know that there exists an extremely robust response to the endogenous retrovirus argument for common descent. Proponents of evolution publicize ERVs as irrefutable evidence for their position. Some have claimed this is as close to proof as you can get in science. For these reasons, I found the writing of this handbook to be absolutely necessary. As a skeptic of common descent, I knew there must be a comprehensive response to shared endogenous retroviruses between humans and the great apes and other forms of life that satisfactorily answers each of the challenges and questions associated with this topic. And that is exactly what this book is: an extremely comprehensive examination of endogenous retroviruses as evidence for common descent that accomplishes exactly what I set out to do, which is to answer every major challenge on this topic. To my delight, it turned out that encountering endogenous retroviruses or I should say, the misrepresentation of ERVs, I discovered one of the most powerful and persuasive arguments for separate ancestry. This book is essentially the culmination of years worth of research on the topic of endogenous retroviruses and ancestry in general. I have debated the best the evolution camp has to offer, and I have seen firsthand their greatest so-called arguments. I am here to tell you confidently that their best arguments have all failed. This book is loaded with sources and data that will leave the reader astounded with how remarkably the scientific evidence refutes common ancestry and confirms biblical creation. Every reference is thoroughly documented. The reader will be pleased to discover countless technical papers supporting nearly every single one of my arguments presented. The more I investigated the primary source data, the more I realized just how bankrupt common descent was. It is interesting that what the evolutionary community often claims is their best evidence for common descent actually turns out to be some of the greatest evidence for separate ancestry. The verdict is in, and common ancestry has been invalidated. Endogenous retroviruses are not good evidence for evolution, and the evidence for separate ancestry is undeniable. The militant critics of biblical creation will fail to counter the arguments offered in this book, and many have already responded by ignorantly claiming if it isn't in the primary literature, that is the arguments in this book, then why should they care? Well, it must feel unfortunate for the defenders of common descent to know that it is the primary literature itself that has thoroughly refuted ERVs as evidence for evolution. The evolutionary community has put forth their best arguments, and this book is about to demonstrate exactly why these arguments have all failed when examined. And that concludes the first part of this book, the preface. And after this section, we get into the introduction, and we'll save the introduction for the next episode, this series. I would like to go over a few things, give a little commentary, though, before we wrap up this episode. And that has to do with a question that you'll see asked several times throughout this book. It's an important question. It's a question that I frequently ask evolutionists in debates, in discussions, in impromptu open mic debates. And that question is, are endogenous retroviruses really the ancient remnants of past viral infections, or are they created units of DNA function? And the answer to that question has incredible implications for the origins debate. Well, as you're going to find out throughout this book, endogenous retroviruses are far from being junk DNA. What we find in terms of the signature of what would be expected based on that question, again, are these really the ancient remnants of past viral infections, or are they created units of DNA function? The answer to that question is based on what we find in the conventional literature itself. We find important or significant function after function associated with ERVs. You're going to find all throughout this book plenty of literature, right from the conventional literature itself, that demonstrates ERVs are functional in placental development, in embryological development in general. They're functional in the immune system, cell stress responses, determining cell types, gene regulation, gene expression. Gene expression, the important process by which a gene is turned on in a cell to make RNA and proteins, right? You got DNA, RNA, and protein synthesis. And so endogenous retroviruses and other classes of retrotransposons are involved in these important aspects of DNA function, DNA regulation, protein synthesis. As a matter of fact, without endogenous retroviruses, the placenta, and I talk about this in great detail throughout the book, the placenta, as we know, it's critical to pregnancy and embryological development. The placenta would not actually work. We would not exist. There's one paper that I love, and you'll see again this paper mentioned throughout this book, where they talk about how if the accidental infection of a mammalian ancestor, there's their presupposition or assumptions, evolutionary assumptions, did not occur, we would say this is a created unit of DNA function. So there wasn't an exogenous retrovirus infection in the past that became endogenized. No, this is a created unit of DNA function. So it makes sense that mammals wouldn't exist without it. That's what this specific paper talks about, that mammals would not exist if we didn't have ERVs. I like to exist, and I hope the reader or the listener likes to exist. So basically, the question comes down to, why do various organisms share endogenous retroviruses? I wouldn't be surprised, and I'm not surprised to find out that the many forms of life share endogenous retroviruses. It's like asking the question, why do all kinds of modes of transportation share engines in common? Well, guess what? In the same way that without many of these retrotransposons, we wouldn't exist. Without an engine, the car wouldn't run. The car basically wouldn't exist because it doesn't have an engine to actually do anything other than sit there and, be something that you look at. And so why do GMs and why do Hondas and Chryslers share engines in common? It's because of common design. It's as simple as that. Now, why these ERVs form nested hierarchical patterns, for example, we're going to get into that in great detail, but I did want to point out that many of these endogenous retroviral sequences, these functional stretches of DNA, many reasons why they're there is so we can exist. Like in placental development, they're essential for life. And not only that, ERVs don't just assist in embryological development and embryonic immune systems, okay, because endogenous retroviruses actually help safeguard the baby from viral infections. So as the baby's growing, endogenous retroviruses are protecting that baby. But as we come out of the womb as a baby and, we're born into this world and we grow up and we get older, well, what happens is these endogenous retroviruses actually work in our immune systems during those various stages. They act greatly in the immune system. They're like antiviral protectors in the genome. Think of an antiviral system in your computer. And so our immune system pathways are actually dependent upon enzymes generated by, guess what? Endogenous retroviruses and ERV-like elements. We literally have these classes of retrotransposons in our genome that are safeguarding our bodies against microbes and other viruses. And so they are engaging, they are participating and allowing for small scale, but very important changes in how the human body works in general. And so is it a surprise, again, that we actually share endogenous retroviruses with other forms of life? No, it is not surprising. As a matter of fact, the existence of endogenous retroviruses, they play roles in determining cell types, gene regulation, gene expression, placental, and embryological development. They're active in the immune system. The reason why we share these, actually the fact that we share these, is confirmation of separate ancestry and intelligent design. And oftentimes, and you'll see this throughout the book in detail, but just to get everybody as excited as I am about this topic, as you can tell, I'm excited. I am excited, and I want to make this technical topic interesting to everybody. I want everybody to be pumped up. I want everybody to be fired up about this topic. The evolutionary community, they think that endogenous retroviruses are irrefutable evidence for common descent, for why they believe that they're related to a strawberry. I'm here to tell you that endogenous retroviruses are inconsistent with common descent and act as confirmation for separate ancestry. Well, evolutionists, they'll look to the structure of ERVs. And they'll look to the sequence similarity that ERVs, endogenous retroviruses, meaning endogenous occurring from within, that they have to exogenous retroviruses. So exogenous retroviruses occurring from basically outside. Think of the HIV virus, right? The HIV virus is passed on through horizontal infection from one person to another, or an exogenous retrovirus being passed on from one organism to another. The endogenous retrovirus occurs from within. It exists in our genome. And so the theory goes that in the past, exogenous retroviruses bombarded organisms and they invaded in the right way. The right way would be the germ cell line, right? Sperm or egg cell, reproductive cells, in order that they can be passed on, not horizontally, but vertically. And so when they're passed on vertically, every single cell of the progeny has that viral DNA. And so evolutionists will ask the question, why did God therefore create living organisms with viral DNA? Well, firstly, and I got a whole section on this in the book, this goes back to the question regarding the origin of retroviruses in general. And I like to ask, what came first, the retrovirus or the host? Because retroviruses require hosts to replicate. And so without a host, there's no replication. There's no retroviruses. And so what I put forth is called the escapee hypothesis, very consistent with what many virologists in the secular world agree with as well, because there's a chicken and egg problem for the evolutionists, especially if they want to hold to a virus first hypothesis. And so as creationists, we can say that exogenous retroviruses, harmful exogenous retroviruses, occurred or originated from healthy host DNA. And from there, they can then be transmitted or cross species from one species to another. And when they cross a species and its original host to a novel host, the host doesn't know how to regulate that invading virus. And so it burns hot and fast. And then you have a harmful virus. Okay. So the evolutionists have a backwards question. They need to ask, where did exogenous retroviruses come from to begin with? They also need to ask themselves if they can provide evidence. But if they don't want to ask themselves this question, I have no problem asking it for them. Can they provide even a single example of an exogenous or harmful retrovirus infiltrating the genome of a living organism, infiltrating in the right way, and so in a germ cell line, being passed on? The harmful effects of that retrovirus are done away with through mutations, and then they evolve the ability to basically be essential for life to exist. Have we ever seen a non-functioning endogenous retrovirus going from non-functioning or beneficial to the host to something incredibly functional in embryological development or determining cell types? No. And as a matter of fact, many of the functional roles associated with ERVs, as we're going to see throughout this book in detail, I'm just kind of covering a few things in a nutshell. Many of their functional roles require their sequence similarities to exogenous retroviruses. They require the GAG, the POL, the ENV components of the ERV. They require the LTRs, the long terminal repeats, to carry out their functional roles. One of their more amazing functional roles has to do with viral mimicry. And so basically, what this viral mimicry does is it helps with tumor suppression, fighting off tumors. This is why researchers are actually working on drugs that will stimulate what's called the p53 protein. And so they're looking to stimulate p53 activity. This is basically the superhero or guardian of the genome. This is how important this DNA element is, okay? And basically, when the cell is under stress, transcribed ERV elements, created ERV elements, they give, because they have that sequence similarity to exogenous retroviruses, they give the cell the appearance of basically being invaded by a viral infection, being invaded by an outside attacker. And what this results in is the targeting of the cell more. Basically, the cell is targeted more destruction by the immune system. So you have the immune system attacking the cell because the cell is giving off the appearance of being invaded by a viral infection. But what actually is happening is these tumor cells, they're being eliminated, they're being destroyed. And again, this whole mechanism is mediated by that p53 protein. And so this is a way for those harmful tumor cells to become detectable by the immune system. And that enables the amazingly designed immune system to clear these tumor cells from the body. But in order to carry out this role in viral mimicry, the ERVs have to look like harmful exogenous retroviruses. It's akin to an FBI agent who wants to infiltrate some criminal gang or the mob. Well, he can't show up in his infiltration in his FBI uniform with his name badge on. Hey, I'm Mr. Smith. I work for the FBI. That's not going to work. That's not going to last very long. No, he has to play the part. He has to look the part in order that they don't detect who he really is, figure out who he really is. And so these ERVs, it's the same thing. They got to look the part to carry out this role. And so to carry out many of their roles, including fighting off exogenous retroviruses, including viral mimicry and tumor suppression, they have to look like exogenous retroviruses. And it's amazing because, again, this is something we'll be talking about in the book, but you have koalas. Koala bears are being studied, and they are actively being invaded by exogenous retroviruses that are becoming endogenized. Well, it turns out that pre-existing endogenous retroviruses are playing a role in destroying those newly endogenized retroviruses. So you have pre-existing ERVs fighting off these newly formed endogenous retroviruses. They're disrupting their sequence. And this role is also carried out based on their sequence similarity to exogenous retroviruses. And so this is amazing stuff. This is science fiction almost, what we're seeing on a molecular or genomic level. It's a great time to be a young earth creationist. I love studying this topic. I'm really enjoying this active research project on ERVs and showing the evolutionists exactly why ERVs are not good evidence for common descent. And so you're going to see in this book, I do make predictions that come down to the nature of fixed versus unfixed ERV sequences. What that means in genetics and population genetics is that if an ERV is fixed, that means every single person in the population has them. They're stuck in place, basically. Everybody has them, not just a subpopulation. And those are shared with chimpanzees. But those, I would argue, and I do argue in the book, are the created units of DNA function. And therefore, I would predict function for most of them. Of course, we live in a fallen world. Mutations accumulate every single generation. And so mutations can destroy a pre-existing function in an ERV, or a mutation, deleterious mutation, can actually take a healthy functioning ERV, right, that's created into something that's disease causing. Okay, so there's a lot that goes into this. But if we have what's called an unfixed ERV, where not every single person has it, then I predict in this book that those would be the result of true endogenous events. Basically, where an exogenous retrovirus invaded, got passed on through the germ cell lines, and became endogenized. But those would be unfixed. And so we're going to get into that throughout the book. And the gold standard of science is making testable and repeatable predictions that are falsifiable. And that's exactly what I do in this book. You're also going to notice, and here's the last thing I want to say, because I want to keep each episode less than an hour. You'll notice how 90% of these so-called ERV sequences are actually not true endogenous retroviruses. as in they don't resemble the full ERV units as I've been explaining, right? That comprise your LTRs, your GAG, your POL, and ENV genes. Okay, the majority of your so-called ERVs, I'd rather call them created units of DNA function. You know, the ERV did not call itself an ERV. The pseudogene did not call itself a pseudogene. We're kind of stuck with those names, right? But unfortunately, when an evolutionist hears virus, they think of something bad or harmful or disease causing, but that's not the case. And it's just a shame that there's a lot of presuppositions going into what these units are called. In this case, endogenous retroviruses. But you'll find that your what's called solo LTRs, you find thousands of those, okay? Incomplete ERVs. We understand now that they simply represent functional stretches of DNA. These solo LTRs, these long terminal repeats, they're actually serving as binding sites for initiating or enhancing gene expression. Gene expression, again, is that process in the cell in which a gene gets turned on to make RNA and proteins. This is important to the function and life of an organism. And these solo LTRs, which evolutionists assume are degenerated ERVs, again, they're serving as binding sites for initiating this process or even enhancing this process. And so when you hear evolutionists say, "Oh, we share thousands of these with primates," that's not true, because 90% of those so-called ERV sequences are assumed to be ERV sequences when in fact they're just solo LTRs. They may not even be ERVs. As a matter of fact, they're probably not ERVs. But it's amazing stuff, guys. ERVs can literally act as promoters. They can act as enhancers. Again, they can act as transcription start sites for gene expression. We understand proteins encoded by these endogenous retroviral sequences heighten or magnify. They amplify immune responses through the binding of very particular receptors. So every part of the ERV is functional and necessary for the ERV to carry out its amazing roles. The proteins associated with ERVs, guess what? They actually interfere or disrupt incoming viral particles. Your envelope proteins, right? Your ENV component of the endogenous retrovirus, they have an operational role in blocking harmful viruses from entering the cell. And so these harmful viruses, they want to enter the cell of the host. They want to cause damage. They want to cause disease. They want to take over. But these ENVs, these envelope proteins, they actually bind to receptors. Okay. And this disallows the infectious viruses from entering. This is amazing evidence for design. The fact that these viruses are inactivated before they can infect new cells speaks to forward thinking. And forward thinking mechanisms in the genome point us back to what? The forward thinker who front loaded organisms with these amazingly intricate and well-designed systems. And so, guys, you're in for a wild ride. This walkthrough is very important. I am close to the hour mark, so I am going to wrap it up. Again, this is just part one. I'm just giving you a taste of what's to come on this topic because I want you guys to be as fired up. I want you guys to be as excited as I am about this topic. This topic being what? Endogenous retroviruses. And so with that, everybody, we are going to wrap it up. Stay tuned for part two, which should be available very shortly. This information is important, guys. Share it around and please consider becoming a Standing for Truth Ministries partner. God bless all. Donny's out.